Okay, so today we are going to discuss polysaccharides. Now, polysaccharide, the name only it says that it must contain number of monosaccharides, large number of monosaccharides. So, polysaccharides that have large number of monosaccharides together by glycosylic linkage. Now, there are few examples of polysaccharide. The first one is starch. Now, you must have heard about that starch. It is the main food storage in any living body. It is found in cereals, roots, some vegetables, etc. The polymer it is the polymer of alpha glucose, means number of alpha glucose connected with each other by glycosidic linkage to make this starch structure. Now, this is the monomer of alpha glucose, that is the monomer of starch. But this monomer, they are connected in two different manner in the structure of starch. Again, alpha glucose, they are connected with each other in two different manner in the structure of starch. Now, that two different manner leads us two main components of starch structure. It is not monomer. These both are amylose and amylopectin. They both are two components of starch. The monomer is alpha glucose. Starch is polysaccharide, in that the monomer is alpha glucose. These two are the components. Why these two components are there? Because alpha glucose connected with each other in two different manner. So, amylose, the first component, that is soluble in water and around 15 to 20% present in starch. Amylose is made up of alpha D plus glucose, and the number that is 200, 2000 or 400. Now, they are connected with each other with C1 to C4 glycosidic linkage. Structure we will see here. While amylopectin, the second component, that is insoluble in water. You can see amylose, that is soluble, and amylopectin, that is insoluble in water. Amylopectin is 80 to 85% present in starch. In amylopectin, the alpha D plus glucose, they are connected in branch manner. Here in amylose, alpha D plus glucose, they are connected with each other in linear manner. But amylopectin has branch structure. You can see this one is amylose, the structure of amylose. This chain continues here, both sides. So it is the linear structure. You can see this one is carbon number 1 and this one is carbon number 4. The C1 to C4 glycosylic linkage is there. While in amylose, this structure is already present. The same structure is here. But with that, this branch is attached. So it is a branch structure. So there are two glycosidic linkage, C1 to C4, this, and second one is this, C1 to C6. Two glycosidic linkage is there in amylopectin. Again, amylose and amylopectin both are component of starch. They both are a polymer of alpha glucose only, but the connection is different here. Amylose, that is linear structure, Amylopectin, that is branch structure. That is the major difference between them. Amylose, that is water soluble. Amylopectin, that is water insoluble. Okay. Now the second polysaccharide, that is cellulose. Now you must have known that in plant cell wall cellulose is present. Now cellulose is made up of beta D glucose. Starch was made up of alpha D glucose. Here beta D glucose, they are connected with each other to form cellulose. Now here the linkage is carbon number 1 of 1 glucose and carbon number 4 of second glucose, means C1 to C4 glycosidic linkage is present. The structure is like this. You can see this one is carbon number 1, this one is carbon number 4. C1 to C4 glycosidic linkage is there. So this is the structure of cellulose. Now important point is there, that is protein. Now what are the sources of protein? Milk, cheese, peanut, fish, meat, etc. They are the main sources of protein. All proteins are made up from alpha amino acids. Now what is amino acid? You can see in the name only it says amino means amino group and acid means carboxylic acid group. Both group must be present in same structure. As example, you can take this. You can see here COOH is there and NH2. Amino group and carboxylic acid group both are in same structure. So this structure is called amino acid. In addition to that, you can see that among these two, COOH is the strong group. And adjacent to that, this carbon is there, so it is our alpha carbon, and on alpha carbon we have NH2, so it is called alpha amino acid. Okay, again COOH, that is our strong group. Adjacent to that group, this CH is there. Now it has NH2 group on it. This CH is called alpha carbon and it has NH2 group, so this structure is called alpha amino acid. Both amine and acid group are present. All amino acid are having trivial names. These names are based on their property or source of the compound. As an example, glycine, that is one of the amino acid, and it is named after its sweet taste. Now in Greek word glycos means sweet, or for that we have this name glycine. Okay. Another example, that is in Greek word tyros means cheese, and from cheese we have derived a amino acid, so we named it after that tyrosine. Okay, it is from the source of the protein. The source is cheese, and in Greek word cheese is called tyros, so from that we named it tyrosine, the amino acid. Next one is classification of amino acid. Now amino acid is having both NH2 and COOH group. So if a structure has more NH2 group than COOH, means number of NH2 group is more than number of COOH group, then the overall nature of the compound is basic nature. So the classification says that it is basic amino acid. If number of COOH group is more than number of NH2 group, then the amino acid is acidic amino acid. And if both are same, COOH and NH2 group are same, then the compound is neutral amino acid. This is the classification of amino acid. Again, number of NH2 group is more, then basic amino acid. If number of COOH group is more, then acidic amino acid, and both are equal, then it is neutral amino acid. Now you can see this one is amino acid, alpha amino acid. In that, this nitrogen that has two non-bonding electron. So what it will do? It will attract H plus from this acidic group. So it forms a resonance structure with resonance structure here. You can see RCH, then NH3. Here hydrogen is from this COOH group. You can see here. So COOH becomes COO negative ion, and NH2 becomes NH3 plus ion. This structure, it has positive and negative ion in same structure. That is called Zwitter ion. Again, the definition is important. If positive and negative ion both are in same structure, that is called Zwitter ion. Positive and negative ion both are in same structure, it is called Zwitter ion. Okay.